Hello students, welcome to another lecture of Biochemistry. Your teacher is back, so you are listening again to your very favorite Dr Sandeepan Sengupta, who is an assistant professor in Microbiology. In our previous classes and lectures, we have been discussing about various aspects of protein structure. We have started from amino acids and we have covered a very long journey going on through different levels of protein structure. We took a very thorough look over 20 naturally occurring amino acids, over the structural features of amino acids, over the special features exemplified by each of these 20 naturally occurring amino acids. Then we came across unusual amino acids or uncommon amino acids which do have very specific and which do have very important roles in biochemistry. Next to that, we studied about peptide bond, we studied about the structural and the geometric features and the biochemical nature of peptide bond. We all know that peptide bond has a partial double bond character and is rigid and coplanar in nature and shows resonance. Now, the basic things are known to you. Now let us move on, continue our discussion with protein structure. Now today's topic of discussion is the quaternary structure of proteins, or the 4-degree structure of proteins, which is the highest order of protein structure. So as you all know, what are proteins? The proteins are nothing but the polymers of amino acids which are linked together by peptide bonds. The peptide bonds are the CONH amide bonds which link the amino acids together and are generated as a result of condensation reaction in between the two neighboring or the participating or the adjacent amino acid residues. Now, if we directly move on to protein structure, there are 4 different levels of protein structure. The fundamentals of the protein, different levels. The primary levels of protein structure are already known to you, and I have discussed about all those things, about the basic issues in all my previous classes and lectures. So there are 4 different levels of protein structure to recapitulate, that is to make you all recall. That is primary structure, secondary structure, tertiary structure and quaternary structure. So primary and secondary structure are the 2 basic levels of protein structure, while tertiary and quaternary structure are considered as the higher orders of protein structure. So what do you mean by primary structure? It is the linear sequence of amino acids, of constituent amino acids of a protein or a polypeptide. And how are the amino acids linked to primary structure? The amino acids are linked by peptide bonds or CONH amide linkages. Now what is secondary structure? Next to primary structure comes the higher order structure of protein, which is called secondary structure. Secondary structure is nothing but refers to the local conformation that is adopted by a part of the protein or polypeptide. So secondary structure describes in itself a structural arrangement, or that is a form of arrangement which is being adopted by a part of the protein or polypeptide. There are several different forms of elements of forms or elements of protein and secondary structure, like alpha helix, the most prevalent form, then beta pleated sheets. Other than these, we have beta turns, gamma turns, etc. Now, in our last class, we ended our discussing all about supersecondary motifs of proteins and about the tertiary structure of proteins. Now, today I am over here to discuss about the 4th or the highest order of protein structure, that is quaternary structure. But before that, after listening to this lecture, you'll be able to answer to a few very fundamental aspects of protein, that is quaternary structure, that it is applicable for which class of proteins or what kind of proteins. Do all proteins have a tertiary quaternary structure, or is it that only a particular or only certain kinds of proteins which meet certain criteria have a quaternary structure? That is a very important question for you all. So let us first find an answer to this, that quaternary structure is, or the highest order of protein structure is applicable applicable for which class of proteins, or for which type of proteins rather, not class, for which type of proteins rather. Now depending upon the number of peptide units and peptide chains and the subunits present, there are a variety of proteins, and we can group proteins into monomeric, oligomeric and multimeric proteins. So what are monomeric proteins? Those are monomeric proteins are the proteins which have a single peptide chain. So the common examples of monomeric proteins are lysozyme, found abundant in egg, then tears, human saliva, then human serum albumin, then other common examples of monomeric proteins include cytochrome C, ribonuclease, etc. Now next to monomeric proteins are oligomeric proteins and multimeric proteins. So what are these proteins? Actually, when a protein doesn't consist of a single peptide chain but consists of, or is a combination of, multiple peptide chains linked together in a compacted structure, it is then called an oligomeric protein or a multimeric protein rather. So a protein with multiple peptide chains is called a multimeric protein, and the simplest type of a multimer is an oligomer, which consists of 8 to 10 different kinds of peptide chains, or which has peptide chains lying in number in between 8 to 10. So that is an oligomeric protein. Now the question arises over here is, that is very important question to be addressed, is that quaternary structure is, or the highest level of protein structure, is actually meant for which type of proteins? Is it applicable for monomeric proteins, or is it applicable for oligomeric and multimeric proteins, or is it applicable for all the three types of proteins with different peptide chains? That is for monomeric, for oligomeric or multimeric. What do you think? So I'll ask you to recall the aspects of tertiary structure of a protein. So what do you mean by tertiary structure? Now I hope you all know that next to secondary structure, several different that is combination of the elements of secondary structure are grouped to form supersecondary motifs, or classes called supersecondary motifs, in which the several elements of secondary structure are combined and grouped together, like combination of alpha helix plus beta sheet, alpha beta strands in which alpha helix and segments of alpha helix and beta sheet are interspersed, etc. Next to this, the supersecondary structure and the adjacent elements of second, along with the neighboring elements of secondary structure, are grouped or are further compacted and folded to form a higher ordered structure, which is called a domain. And there are multiple domains present in every peptide chain of a protein. So now hope it's clear to you that it's already known to you that what do you mean by a domain, and how the hierarchy goes on, starting from secondary structure till tertiary structure. That is, so what is tertiary structure? That is, tertiary structure is nothing but the spatial relationship and the three-dimensional arrangement of domains and the spatial relationship between domains present in a single peptide chain of a protein forms what is called a compact structure called tertiary structure of a protein. So, hence, tertiary structure is applicable for which class of proteins? It is applicable for monomeric proteins. So, for a native monomeric protein, its native structure, or its overall three-dimensional structure, is its tertiary structure. Why? Would you like to ask over here that why is it so? Yes, the answer is known to you all. It is because a monomeric protein has a single peptide chain which contains or which is made up of multiple domains. Hence, all the spatial arrangement and the structure that is being adopted by all these domains of the peptide chain or single peptide chain of monomeric protein forms the tertiary structure of the peptide chain of that monomeric protein, which is itself the structure, the three-dimensional structure of the entire monomeric protein. because the protein has a single peptide chain. So that is what is called a tertiary structure, and it's applicable for monomeric proteins. Now, what do you mean by quaternary structure? It is a still higher order of protein structure when a number of peptide chains are compacted, folded and looped to form a compact three-dimensional structure. That is a compact three-dimensional structure. That is called a tertiary quaternary structure, quaternary structure, or the 4-degree structure of a protein. So it is applicable for which class of proteins? Hope it's now clear to you that quaternary or the 4-degree structure is applicable only for oligomeric and multimeric proteins. The highest order of a monomeric protein is its tertiary or the 3-degree structure. But for multimeric or oligomeric proteins with multiple peptide chains, every peptide chain will have its own 3-degree or tertiary structure, and all these structures of all these peptide chains are further compacted and grouped together, folded and looped to form or to generate a still higher order of structure, what is called a 4-degree or quaternary structure of a protein. So it's applicable for multimeric and oligomeric proteins with having multiple peptide chains, mostly for oligomeric proteins having peptide chains lying in number around 8 or in between 8 to 10. So that is what is called as the quaternary or the 4-degree structure of proteins, which is applicable for not monomeric but is applicable for multimeric, that is oligomeric proteins. Now, what is the arrangement of subunits in such an oligomeric protein? That is, such an oligomeric protein has a number of identical subunits arranged together in its quaternary or 4-degree structure, the highest order of structure, in a symmetric pattern. Or often it is combination of a number of non-identical subunits. But if it has a number of non-identical subunits, that disrupts the symmetry of the protein. So what happens in quaternary or the highest order of protein structure of an oligomeric protein, number of identical subunits or number of non-identical subunits grouped together is repeated several times to form a particular or to form a single unit which represents a structural unit of the quaternary or 4-degree structure of protein of such an oligomeric protein, and is called a protomer. So what is protomer and how is the structure? How does different subunits make up the quaternary structure of an oligomeric protein? It's quite obvious that it has 8 to 10 peptide chains. So the different subunits are either identical, which are arranged in symmetric fashion, or maybe not identical, but then the non-identical subunits are grouped together to form again a similar pattern that is repeated multiple times. And that repeating pattern or structural pattern of the quaternary structure of an oligomeric or a multimeric protein is actually called as a protomer. So I hope the terms oligomeric protein, then quaternary structure, then protomers, these are the important terms, the terms of huge biochemical importance in protein chemistry, or of huge biochemical importance, are quite well clear to you. Now, I leave a very important question for you at this point. I have a very important question for you. Can you think about the different possible ways or the different possible arrangements in which the protomers are arranged to form the quaternary or the highest order of structure of an oligomeric protein? It's a very interesting thing, and for that you really need to peep into you really need to think over the issue. So as a part of your activity, I'm leaving this question for you all. Think about the different types of arrangements, about the symmetry, about the different types of arrangements, and about different symmetrical patterns in which the protomeric subunits of an oligomeric or a multimeric protein can be arranged to form on its highest order of protein structure, so called the quaternary structure of a protein. And I leave another very important question for you all, that what is the importance of quaternary structure of a protein. So keep doing with all these two very important assignments. I'll be resuming back after a very short break. So see you all after a break.